Welcome to the deep dive.
Great to be here.
Today, we're really plunging into the very complex world of therapies for Alzheimer's disease, AD, and Lewy body dementia, LBD.
Yeah, it's a tough area.
And we're not just, glancing at the headlines. We're digging into insights from a recent forum. It was at the Alzheimer's and Parkinson's Diseases Conference.
That's right, ADPD. Top researchers were there, speaking quite openly.
About the hurdles, the challenges, but also the promising future paths in this really critical field.
Exactly.
So think of this as your sort of insider's look. Why is it so hard to create effective treatments for these, devastating diseases?
And what new approaches are people genuinely exploring?
Right. And crucially, why does something that looks good in the lab, in animals, often just fizzle out when you try it in humans?
And what's really striking, I think, is the candiness you mentioned. These experts, they don't sugarcoat the limitations.
No.
The limitations of our current research models, the frustrations we've all seen with translating findings into actual treatments.
So our goal for you today is to look beyond that surface level news.
Yeah, to really understand the intricate layers of this research, appreciate the obstacles, which are significant.
But also the potential, the real potential of these emerging strategies.
Absolutely.
Okay, let's unpack this then. A key point right at the start was this difficulty, this challenge of translating results. You get encouraging data from animal models, and then it just doesn't work in humans.
Yeah.
What's the core reason? Why the disconnect?
That's a really fundamental challenge.
Yeah.
As one researcher put it, our models are, they're inherently simplifications.
Right. Not the real thing.
Not the actual human disease, no, human AD, LBD. These develop so gradually over many, many years.
Decades sometimes.
Decades. And they manifest in such a wide range of ways from person to person. We're really grappling with the sheer complexity of human biology.
Where ideally you'd want precision medicine. Right. Treatments tailored to the individual.
That's the goal. But achieving that is incredibly difficult.
Mm-hmm.
The biology is just so complex.
Right. So the mouse models we often hear about in the news, they're not like perfect replicas of what's happening in a human brain.
Precisely. No. They are carefully designed systems, very useful systems. They help us dissect how specific molecules behave, how they interact within a living organism.
Yeah.
But the danger lies in drawing overly direct conclusions.
Or simplistic ones.
Or simplistic conclusions, yes. Assuming they perfectly mirror the human condition, it's just not the case.
So what's the alternative? Or how do we manage that?
Well, the researchers really emphasize the crucial need to investigate human biology as thoroughly, as ethically, and practically possible.
Even though that's hard.
Even though it takes considerable time and, frankly, expense. Yeah. The core message here for you is that we just can't take positive results in mice at face value.
Need more evidence.
We need to see evidence that these findings truly reflect the much more intricate reality of human disease progression over decades.
It's interesting, one expert made the point that this isn't just an Alzheimer's problem, is it?
No, not at all.
Even in, say, cancer research, animal models rarely, if ever, perfectly mimic the human disease. Why is that analogy relevant here?
Well, it just provides valuable perspective, doesn't it?
Yeah.
It reminds us that we use these models as tools.
Tools to gain insights.
Exactly. But we must always be aware of their inherent limitations. And that's true across all fields of medical research.
Okay. But there are efforts to make better models, right? I think the NIH-funded Model AD program was mentioned.
Yes, absolutely. And this is a really substantial coordinated effort.
Aimed specifically at?
Specifically targeting the creation of more human-relevant models. The approach is multifaceted, layered, you could say.
Layered. How so?
Well, it starts by focusing on genes we know are central drivers of the disease. But importantly, it also incorporates genetic info from GWS studies.
GWS, genome-wide association studies.
That's right. Think of it like detectives looking at the DNA of thousands of people with and without the disease.
To find common genetic clues.
Exactly. Even if those clues aren't the single, smoking gun, they add pieces to the puzzle. And they're also adding in relevant environmental factors.
Like diet.
Like the effects of a high-fat diet, for example.
Yeah.
Things that make the models better reflect the complex interplay of factors in human disease.
So the goal is to build models influenced by more of the genetic mix, more of the environmental stuff we think contributes in people.
Exactly. And a real significant point was raised here. As these models become more like the human disease process,
Yeah.
the disease-like characteristics they develop, the phenotypes, might actually be less severe
Oh, interesting.
and take longer to manifest, which could potentially mirror the slow, progressive nature we see in humans.
But that creates its own challenges for researchers, right? Longer timelines?
It does. Big challenges for study timelines and costs.
No.
But it could ultimately lead to more reliable, more translatable findings.
Makes sense.
And it's also becoming clearer, that no single model can fully represent the entire spectrum of these diseases.
So each model might need to focus on a specific mechanism.
That seems to be the way things are heading. Yes.
Yeah.
Focus on a specific underlying mechanism.
Now that we've kind of explored the limitations, let's dig into those efforts to create better ones, starting with this NIH program.
Sounds good.
One aspect of AD people talk about a lot is how the pathology spreads. Amyloid beta, tau.
Yeah.
It moves through the brain, right?
Yes, that's a key feature.
Are current models good at replicating that spreading process?
Well, this was highlighted as an area where current models often fall short.
How so?
Many models, especially those that rely on introducing foreign genes, transgenes, to induce pathology.
Yeah.
They tend to initiate this pathology more or less simultaneously across many different brain regions at once.
Okay, but that's not how it happens in humans?
Not typically, no. When we look at human PT scans, what we see is a much more gradual, sequential spread of these pathological markers.
Interesting. So the models often just hit the gas pedal right away?
That's a good way to put it. Whereas the human disease follows a more protracted course, region by region, often.
Were there specific models mentioned that do try to capture this spread?
Yes, there was specific mention of models developed, I think by Matthias Jucker and colleagues back in the early 2000s, actually.
And they address this spread?
They do. Models that address this amyloid beta-induced spread of pathology. And these models are particularly relevant, right?
Why, particularly?
Because patients are typically diagnosed after the disease has already begun. This spread is actively occurring.
So you want to test treatments in that context?
Exactly. But the experts noted that these more sophisticated spread models haven't actually been used as widely as they maybe could be.
For testing interventions when the disease is already progressing?
Precisely.
It seems there's this growing awareness. We need models that better mimic the timeline, the way the disease actually unfolds in the human brain. What about the differences between the rare inherited forms of AD or LBD and the much more common sporadic forms? Does that affect the models?
Oh, absolutely. That's a really critical distinction. The experts pointed out they've seen much greater success translating findings for rare monogenic diseases.
Where it's caused by a single gene mutation.
Exactly. Lysosomal storage diseases were given as a prime example.
Okay.
In those cases, replacing the missing enzyme in animal models can very closely replicate what happens therapeutically in humans.
But that doesn't hold for the common forms of AD.
It's much harder. When you try to correct single genetic defects in animal models of complex common diseases like late-onset AD, the underlying biology can diverge quite significantly from what we actually see in humans.
So it really underscores the need for caution.
A lot of caution when interpreting results from those types of single defect models for complex diseases.
So fixing one specific problem in a mouse model of, say, sporadic AD might not tell us that much about how to effectively address the same problem in a person with the disease.
That's basically the core message, yes. The intricate interplay of multiple genetic factors, environmental factors, aging, it just makes it a far more complex puzzle to solve using simplified animal models that focus on just one defect.
We've talked a lot about mice. Are there other organisms, or maybe in vitro systems, lab-based systems, that show promise for modeling these diseases?
Absolutely. There's movement on several fronts. The NIH is also funding a marmoset AD project.
Marmosets, the small primates.
Yes. They're non-human primates, genetically closer to humans than mice are. So they could potentially serve as a more relevant model.
Is that a new initiative?
It's more recent, yes, but there's considerable optimism about its potential to sort of bridge that translational gap we've been talking about.
And what about omics data?
Right. The importance of cross-referencing omics data, that's the comprehensive biological info, like genomics, proteomics, transcriptomics.
Right, comparing the molecular details.
Comparing those details between mouse and human studies, using publicly available NIH databases, that was also strongly emphasized.
So just to clarify for me, transcriptomics is looking at all the message copies from our genes.
Basically, yes. Which genes are active?
Proteomics is the actual worker molecules, the proteins.
Correct. What the genes produce.
And metabolomics is the products and byproducts of all that activity.
You got it. The small molecules involved in metabolism. It's looking at the whole system.
Okay, that helps. So comparing these detailed biological changes in mice to what we see in humans with the disease.
Exactly. And when we're trying to answer specific biological questions, the choice of model becomes even more critical.
How so?
Well, take the immune system, for instance. There are significant differences in how it functions between mice and humans.
Really? How significant?
Well, only about 30% of the adaptive immune pathways and maybe 40% of the innate immune pathways are actually conserved or the same.
Wow, that's not a lot.
It's not. So if researchers are investigating, say, immune-based therapies, they really need to select models where those specific pathways they're interested in are conserved.
Otherwise, the results might not mean anything for humans.
Exactly. You need a reasonable chance of their findings translating. That's a really important point. The model has to be genuinely relevant to the particular biological question you're asking.
What about using actual human tissue and cells in the lab?
Yes. What about that? That's another key area.
These in vitro models.
Right. Human tissue and cell-based models. Things like triculture systems, tetraculture systems, even brain organoids.
The mini brains in a dish.
Exactly, the mini brains. They're incredibly valuable tools for studying disease mechanisms at a cellular level, much closer to human cells, obviously.
But they have limitations, too.
Oh, definitely. It's important to remember they are still simplified systems. They're not a whole brain in a living organism.
And their responses can vary.
They can vary depending on, say, the specific stage of the disease being studied or the exact conditions in the dish. Plus, animal models tend to be more genetically homogeneous, right? We breed them to be similar.
Compared to people.
Compared to the vast heterogeneity we see in human populations.
Hmm.
So the overall message wasn't really to dismiss animal models entirely,
True.
but to acknowledge their limitations, keep refining them, and use them alongside these increasingly sophisticated in vitro approaches. They're complementary.
It really sounds like matching the model to the specific aspect of human pathology you want to study is absolutely critical.
Couldn't agree more.
Like if problems with how brain cells communicate synaptic dysfunction, if that's a major feature in human AD.
Then the models should ideally show that too. Precisely the point.
Right.
Early on, the success of amyloid targeting antibodies in mice, reducing plaque levels that did predict a similar effect in humans, plaque reduction happened.
But that wasn't the whole story.
No, because the initial mouse studies often didn't thoroughly examine synaptic function or cognition, which are the ultimate clinical outcomes we actually care about in people.
So there was a disconnect between the marker they measured in mice and what really mattered for patients.
Exactly. It highlights that potential gap between what we measure as a win in preclinical models and what truly matters for human health and well-being.
And then there's the massive factor of age. You mentioned this earlier. Late onset AD usually hits neurons that are 60, 70, 80 years old. Whereas mouse models often use animals that are, 6 months old? Equivalent to young adults.
That's right. The huge mismatch.
How substantial a challenge is this age mismatch? I mean, it just blows my mind thinking about how different a young mouse brain is from a human brain that's been working for decades.
It's a very, very significant challenge, no question.
What are the biggest biological differences that gap introduces, and how are researchers trying to get around this fundamental hurdle?
Well, the differences are vast changes in metabolism, inflammation, cellular repair mechanisms, the accumulation of damage over time. But there is encouraging evidence.
Oh, yeah.
That older mice, say, around 16 to 22 months old, which is getting into mouse old age, they can actually show more relevant disease-like characteristics.
Like what?
Like loss of synaptic connections in the hippocampus, which is key for memory, cognitive decline, changes in microglia, the brain's immune cells, other immune system changes too.
So using older mice helps.
It provides a more relevant window into the disease process, yes. But the practical difficulties are significant.
Time and money.
Extended time, much higher costs associated with studying older animals. But it's being done more. And then there's the really exciting development of xenotransplantation.
Xenotransplantation?
Yeah, transplanting human cells, like human microglia, into mouse models.
So you get human cells reacting within a living mouse brain environment?
Exactly. It offers a really promising avenue for studying human cellular mechanisms within a living system, overcoming some of the species differences.
So moving back to the lab dish for a moment, we're seeing more advanced in vitro models that can incorporate genetic factors, study how different brain cells interact. What makes these particularly exciting?
Well, they allow us to investigate disease mechanisms in a context that's, more directly relevant to human biology, at least at the cellular level.
Right, using human cells.
Using human cells, often patient-derived cells with specific genetic backgrounds. Now, sure, the specific modifications or treatments we apply in vitro might seem a bit artificial sometimes.
Because it's not a whole organism.
Exactly. But they're essential for us to understand how cells respond to different stimuli, different stresses, potential drugs. The crucial step, though, is to carefully and thoughtfully relate these in vitro findings back to the complex reality of human biology.
Even if there are differences between the dish and the brain.
Even acknowledging those potential disconnects. The consensus among the experts really was that both mouse models and in vitro models are incredibly valuable.
Not either.
Not either. They should be viewed as complementary tools in the research arsenal. Because if you see consistent results across different model systems,
Yeah, that gives you more confidence.
much greater confidence in potential drug candidates before you move into those really expensive and time-consuming clinical trials.
This idea of triangulation, comparing data from human disease, from in vivo animal models and in vitro lab models, that sounds like a really powerful approach.
It absolutely is.
Yeah.
Because even though each of these systems is fundamentally different,
Right.
finding consistent patterns, say in their transcriptomic profiles, the patterns of gene activity, they can be very informative.
So if you see the same biological signal popping up in multiple places.
Exactly. Like maybe a change in a specific biomarker, say pTau217. If you see that in human data and in relevant animal models and in cell-based systems,
That strengthens the case.
that kind of orthogonal reinforcement, as they called it, significantly strengthens the evidence. It's like each model gives you different pieces of the puzzle
And you're trying to see how they fit together.
to get a more complete picture, And the models themselves are getting more sophisticated too.
Like the humanized mice.
Right. Mice carrying human versions of key genes like APP, MAPT for tau, APOE, and now studying combinations how these human genes interact in the mouse.
Like replacing the whole mouse tau gene with the human one.
Exactly. For example, replacing the entire mouse MAPT gene with the human MAPT gene to investigate its effects on things like lipid metabolism and tau pathology in a more human-relevant genetic context. It's a big step forward.
Okay, with all this increasing complexity, you've got more diverse models, different organisms, just this massive amount of biological data, the omics data.
Yeah, it's exploding.
It feels like we need some way to effectively analyze and interpret it all, which brings us to artificial intelligence, AI, right?
Exactly. AI is increasingly seen as, well, almost an indispensable tool now.
To handle the data flood.
To handle the vast quantities of omics data we're generating. We're almost at a point of information overload, honestly. And AI offers the potential to sift through these multi-layered data sets.
From all the different models.
From various models, to pinpoint the most relevant biological pathways, identify promising therapeutic targets.
Can it help with drug design, too?
It can assist in screening existing drugs, repurposing them, and even designing entirely new drug molecules based on structural data or desired properties.
But it's not a magic bullet.
No, the experts were clear on that. It's a powerful enabler, but it's not a magic solution on its own. The quality and relevance of the data we feed into AI systems, absolutely critical.
Garbage in, garbage out.
The old saying still very much applies. You need high-quality, relevant data for AI.
Complex data that maybe a human researcher might miss.
Precisely. And one particularly interesting application being explored is what they're calling vertical integration using AI.
Vertical integration?
Yeah, combining different types of data, everything from large-scale population studies, epidemiology, and clinical trial results, all the way down to preclinical model data and cell culture findings, trying to integrate it all with AI to identify key points where we might be able to intervene effectively in the disease process.
Wow. Okay, let's shift focus slightly now, back to specific model types. Non-human primates. You mentioned marmosets earlier. What are the main advantages and disadvantages there?
Right. Non-human primates, particularly older ones, have this unique characteristic. They can spontaneously develop some key hallmarks of AD.
Without genetic modification.
Without needing genetic manipulation. Things like amyloid plaques, cerebral amyloid angiopathy, CAA. This makes their physiology inherently more similar to humans.
Especially certain species.
Especially old world monkeys. New World monkeys, like the marmosets, might offer slightly faster research timelines because they have shorter lifespans, but they still develop similar pathologies.
But there are downsides.
Oh, significant hurdles. One is just the limited availability of aged primates. They take a long time to grow old.
And ethical concerns.
Important ethical considerations surrounding their use in research, absolutely, and the very high cost of maintaining these colonies.
So what are they mostly used for now?
Currently, they're primarily used in the later stages of drug development, mainly for assessing pharmacokinetics, how drugs are distributed in the body, and pharmacodynamics, testing effectiveness before moving to human trials.
And the Marmoset AD platform.
That aims to make this type of research more accessible, to allow for a more direct translation of preclinical findings, say from mouse studies, to these more human-like primate models, and to facilitate earlier drug testing in them.
So they offer a system that's biologically closer, but with big practical and ethical challenges. What about the brain organoids? Are they an alternative or complementary?
More complementary, I'd say. Organoids, these 3D lab-grown clusters of brain cells, they can help address some of the scalability issues you have with non-human primates. You can make many more organoids.
Right.
The experts really see both organoids and non-human primates as important complementary tools, different but valuable pieces of the puzzle.
So primates are better for some things, organoids for others.
Exactly. Larger animal models like primates are advantageous for studying things like drug distribution within specific brain regions. That's hard to do in an organoid.
Makes sense.
Organoids, on the other hand, are really useful for generating specific brain regions of interest, like the substantia nigra, which is key in LBD and Parkinson's, and for observing cellular phenomena you might not see in traditional 2D cell cultures.
Okay. We spent a lot of time on the models themselves, but ultimately the goal is improving things for people, right? Behavior, cognitive function.
Absolutely. That's the endpoint that matters.
How well do our current animal models capture these critical aspects? Is mouse behavior really relevant to human cognition?
That is a really critical challenge. There's often, well, a disconnect, a gap between what we can measure at the synapse level, the connections between brain cells and preclinical models, and the actual behavioral outcomes we observe.
So you can fix the synapse but not see a change in behavior?
Sometimes, yes. Or the behavioral tests we use in mice might not accurately reflect the complex cognitive functions affected in humans. But there are exciting technological advances.
Like what?
Things like multiphoton microscopy. This allows us to directly visualize and measure the activity of neural networks, even individual synapses, in the brains of living, behaving mice.
While they're doing tasks.
Exactly. These tools are helping us bridge that gap. The hope is, by making our preclinical assessments of brain network function and behavior more robust, more directly linked to underlying synaptic changes.
You improve the predictiveness.
We hope to improve how well these models can predict human outcomes, yes. And advances in neuropsychological circuit research, understanding the brain circuits underlying specific behaviors, are also contributing significantly here.
So the key is linking the different levels.
Finding ways to connect the high-precision data we can get from animal studies with the perhaps less precise but clinically relevant measures we use in humans, like resting state fMRI or PE scans, making that translation stronger.
Okay, let's pivot now to the therapeutic targets themselves. The forum talked about receptor and inflammation-based targets, but it sounds like the discussion went broader.
It did, yeah.
What are some of the promising new directions in actually identifying what to target with a potential treatment?
Well, one area generating a lot of excitement involves what are being called deep biology targets.
Deep biology?
Yeah, think of them as molecular switches, master regulators that have effects on multiple downstream pathways.
Okay.
Particularly pathways involved in maintaining healthy synaptic function. The strategy here is maybe intervening earlier in the disease process by targeting these fundamental mechanisms of cellular health rather than waiting for damage to accumulate.
And genetics still plays a role.
Of course. Genetics remains a crucial area for identifying potential targets. Yeah. But the experts emphasize needing to use genetic information more intelligently.
How so?
Distinguishing between genes that primarily increase someone's risk of developing the disease.
Like APOE4.
Like APOE4.
Yes.
And genes that are more directly involved in driving the disease progression once it starts. It's a subtle but important difference for targeting.
And timing is key.
The timing of any intervention is absolutely critical. When do you hit the target? Interestingly, though, there are examples like the GLP-1 receptor agonists.
Used for diabetes.
Right. They show some effectiveness in early AD trials, despite not having super strong direct genetic links to Alzheimer's risk itself.
So we need to look beyond just genetics.
It highlights the importance of looking beyond genetics alone when identifying potential therapeutic avenues. The overarching message, really, was the critical need for a much, much deeper biological understanding of what actually causes Alzheimer's disease.
Especially why neurons die.
Particularly the fundamental mechanisms that lead to the death of neurons. But they still don't fully understand that. And without that understanding, identifying truly effective targets is incredibly hard.
The Treat AD Consortium came up. Their mission is finding novel targets.
Yes. The Treat AD Consortium is specifically focused on discovering novel therapeutic targets.
What's their basic approach? How do they do it?
They employ a really powerful integrative omics approach. So they combine information from genetics, transcriptomics, proteomics, metabolomics.
All the
omics, yeah. And they use advanced statistical methods to analyze these huge data sets. A key focus for them is studying the very early stages of the disease, using data derived directly from human studies, human brains, human samples.
And then they test their findings.
Then findings from these big omics analyses are applied to preclinical models, mice, cells for what's called orthogonal validation.
Checking if the patterns hold up elsewhere.
Essentially, yes. Testing if the same biological signals or pathways are relevant in different model systems. The hope is that this rigorous data-driven strategy will identify more relevant targets for future human trials.
It sounds like a pipeline.
It is. Their process involves this continuous cycle. Identify potential target candidates from the human data, critically evaluate their biological plausibility, prioritize the most promising ones based on all the evidence, and then develop tools to specifically engage those targets in the preclinical models.
Did any particular category of targets emerge as a favorite? Immune, neuronal?
Interestingly, no. There wasn't one particular category like immune-related or neuron-specific or vascular that emerged as a clear frontrunner.
What does that tell us?
It probably just reflects the complex, multifaceted nature of these diseases. There are likely multiple contributing factors, multiple pathways involved.
It sounds like they're casting a really wide net with these powerful data tools and then very carefully filtering the results to find the best leads.
That's a good description of the process, yes.
One expert raised a fascinating point. The fact that the disease, the pathology, can be at different stages in different regions of a single patient's brain.
Right. That complexity within an individual.
How does that impact the challenge of finding effective targets?
That's a really crucial consideration because it means different brain regions, even within the same person at the same time, can be at varying stages of disease progression. And therefore, the dominant underlying mechanisms might also differ from region to region.
So one area might be dominated by amyloid, another by inflammation?
Exactly. You could have areas where protein aggregation is the main issue, others where the immune response is hyperactive or metabolic breakdown is prominent, or tau pathology is taking off.
What does that imply for treatment?
Well, it strongly suggests that maybe a single magic bullet target might not be sufficient for everyone or even for all stages within one person.
So maybe combination therapies.
It implies we might need to consider a combination of targets. And think about the potential interactions between drugs that address these different pathological processes simultaneously, or drugs that work broadly.
It's almost like this mosaic of disease activity within one person's brain.
A mosaic is a good way to think about it.
This brings up an interesting idea. Maybe we need to rethink what a therapeutic target even is. Move beyond just the obvious things like amyloid plaques.
Exactly. The thinking in the field is definitely evolving, moving towards identifying more fundamental biological mechanisms.
Mechanisms that could affect multiple aspects of the degeneration.
Right. Rather than solely focusing on targeting a single macroscopic feature of the disease, like a plaque or a tangle, what's the underlying process driving that?
This leads nicely into the idea of multiplex approaches, hitting multiple targets or mechanisms at once. What are the thoughts on combination therapies using multiple drugs versus maybe developing a single drug that hits multiple targets or has multiple beneficial effects?
Yeah, there's definitely growing interest in combination therapies.
Why now?
It's largely driven by the, well, let's say modest clinical effects observed so far with single target drugs, like the amyloid targeting antibodies. They help, but they're not cures.
So people are thinking we need to hit more pathways.
Right. But there are essentially two main ways to do that, as you said. Combine different drugs, each targeting a specific pathway.
Okay.
Or develop a single drug that acts like one of those molecular switches we talked about. influencing multiple downstream pathways all at once.
Is there a preference?
Generally, the latter approach, a single drug with multiple effects, is often preferred by researchers if possible.
Why is that?
Just due to reduced complexity. Developing, testing, and getting approval for combination therapies, especially early on when our biological understanding might be incomplete, can be really challenging from a clinical development perspective.
Makes sense. The audience had a really good question about some of the best-known genes, APOE, APP, PSEN1. Why weren't they initially discussed as new targets? What's the current thinking on these big players?
That's a fair question. The primary reason they probably weren't highlighted as new likely stems from our still incomplete biological understanding of exactly how they contribute, particularly APO.
Even though we know APOE4 is the biggest genetic risk factor for late-onset AD.
Exactly. Despite its very strong genetic association, we don't yet fully understand the precise reasons why the APOE4 variant increases risk so much, while the APOE2 variant seems protective.
And without that mechanistic insight.
Directly targeting APOE therapeutically becomes very complex. Right. What aspect of APOE do you target? How? Without causing unintended problems, there's also active exploration of systemic APOE.
From the liver.
Right. APOE is highly expressed in the liver. Could targeting it there has effects on the brain? That's being looked at. The general consensus seemed to be that even these well-known targets should be subjected to the newer, more rigorous omics-based evaluations we discussed to reassess their potential and refine our understanding of their role.
One expert highlighted GLP-1 receptor agonists, those diabetes drugs, as an example of multifaceted drugs hitting multiple pathways. What's the significance of that for AD research?
It's really encouraging, actually. It serves as an example of how drugs develop for one condition, in this case, diabetes, affecting glucose metabolism, inflammation, maybe vascular health, can show promising results in neurodegenerative conditions like Alzheimer's, even if we don't fully understand the exact brain mechanisms yet.
So we can learn from drugs with broad biological effects?
Potentially, yes. It suggests looking at pathways that might bridge metabolic health and brain health. And related to that, the positive results from a trial of a BTK inhibitor.
In multiple sclerosis.
Yes, in MS,
Sure.
Another neuroinflammatory disease. That trial showed that targeting inflammation both in the periphery, the body's immune system, and potentially in the brain could lead to clinical benefits.
So maybe systemic inflammation matters in AD too.
It strongly suggests the relevance of targeting systemic inflammation or immune pathways could be beneficial in neurodegenerative conditions beyond just MS. The brain and body aren't isolated.
The structure of the amyloid protein itself came up. How critical is structure-based drug design if the amyloid structures in mouse models might not perfectly match human ones?
Well, the fundamental takeaway there was that a deep understanding of the biology is absolutely crucial. More crucial than just the static structure sometimes.
How so?
Look at amyloid beta antibodies. It took several attempts, several iterations to identify the specific parts of the protein, the epitopes that were the most effective and safest to target. That came from understanding the biology, not just the structure.
And the same might apply to tau.
A similar process of detailed biological understanding will likely be needed for other targets, like tau. You need specificity based on biological relevance. And for really complex targets, like maybe prenalamine.
What's that?
It's related to APP processing. The point is, its function likely goes way beyond just releasing amyloid beta. There's probably important intracellular signaling involved.
So just targeting the structure isn't enough?
You have to understand its full biological context and function. Simply looking at a static protein structure might miss crucial aspects needed for effective drug design.
The panel also talked about things they felt might be getting overlooked currently. What key issues or potential targets did they suggest need more attention?
Yeah, several important points came up. One was the significant influence of midlife factors.
Like exercise, obesity.
Exactly. Physical exercise, peripheral obesity, how these moderate the risk of developing AD later, and their likely effects first on the periphery, the rest of the body, and then indirectly on the brain.
And we don't study that connection enough.
There's a recognized need for more experimental studies directly investigating these peripheral brain connections. For example, looking at whether factors released by muscles during exercise might actually act as neurotrophic factors supporting brain health.
Interesting. What else?
The lack of comprehensive biological analyses across all organs and in peripheral blood in animal models was noted. We tend to focus just on the brain.
But the body is connected.
Right. Another major point was the selective vulnerability of specific neuron types.
Like cholinergic neurons.
Cholinergic neurons, noradrenergic neurons. We know they're hit early and hard in AD. It's fundamental to break staging, our understanding of pathology spread.
But it's not studied enough.
There was a feeling it's an area that isn't receiving as much direct research focus as it perhaps should. Why are these neurons so vulnerable? We don't really know.
And research might be too focused elsewhere.
There's a feeling that current research, particularly on amyloid and tau epitopes, might be somewhat biased towards the cortex and perhaps disconnected from the fundamental biology of these selectively vulnerable neuron types early in the disease.
Is there hope for connecting these fields again?
Yes. The hope is that newer technologies like single-cell transcriptomics, looking at gene activity in individual cells, and comprehensive whole-brain atlases can help reintegrate these different aspects of the disease.
Finally, the experts got a fascinating question. If you could develop one new model, what would it be? To really advance the field, the answers were pretty diverse.
They were indeed. Really insightful. Several researchers jumped on the idea of studying resilient populations.
People with pathology but no symptoms.
Exactly. Individuals who have the biological signs of AD in their brains, plaques, maybe tangles, but don't develop cognitive impairment. We need models to uncover the protective mechanisms at play there. Others suggested focusing on a model specifically targeting cholinergic neurons, reflecting their early vulnerability.
Makes sense.
The need for improved models of innate immunity in the brain came up, and even a model specifically designed to study the concept of resilience itself. How does resilience work biologically?
An APOE again?
Yes, a better model for understanding APOE function in the brain, particularly nailing down the key differences between the risky APOE4 and the protective APO2 variants. That's still a huge gap.
Immunity was mentioned again, too.
A more comprehensive model encompassing the entire immune system, innate and adaptive immunity, and also the effects of immunosenescence, the aging of the immune system.
And one last one.
Sex differences. Several experts underscore the necessity of incorporating sex differences in disease progression and treatment responses much more effectively into our research models. We know there are differences, but models often don't reflect them well.
Okay, so to try and wrap up this really in-depth exploration, the journey towards effective therapies for Alzheimer's and Lewy body dementia is, well, it's extraordinarily challenging.
No doubt about it.
Marked by these complexities of modeling human disease in the lab, the need for more human-relevant in vitro systems, the hurdles in translating preclinical findings to the clinic.
It's tough. But it's also clear, I think, that the field is making significant strides forward. Through more sophisticated models, these integrative omics approaches, a deeper biological understanding, exploring novel targets.
For me, several aha moments really stand out from this. The clear explanations for why animal models sometimes fail, that was illuminating.
Yeah, understanding the limitations is key.
And the exciting potential of AI, but also these non-traditional models, marmosets, advanced organoids.
And the shift towards understanding deep biology, resilience, not just pathology.
Right, and recognizing systemic factors, that brain-body connection and the selective vulnerability of certain neurons, it feels like a broader perspective is emerging.
I agree.
Now, it's certainly a lot to digest. If you listening are feeling a bit overwhelmed by all the complexity, that's completely understandable.
That's complex science.
But I think the dedication, the ingenuity, the collaborative spirit of these researchers we've been discussing, it offers a genuine sense of hope.
Definitely.
And having a deeper understanding of these multifaceted challenges is just vital, isn't it? For developing a more informed perspective on this critically important research.
Absolutely, and maybe it raises a really important question for you to consider.
Go on.
Given this increasing emphasis on resilience and the influence of peripheral factors on brain health, could future therapeutic strategies maybe shift?
Shift how?
Shift their primary focus towards enhancing the body's own natural protective mechanisms and bolstering those midlife lifestyle factors that seem to build resilience against neurodegeneration.
Instead of only relying on traditional drugs targeting specific pathologies once they've started?
Exactly. Could we focus more on prevention and resilience building, informed by this deeper biological understanding? It's something to think about.
That's a truly fascinating thought to end on. If this deep dive has sparked your curiosity and you'd like to learn more,
We definitely encourage you to explore further.
maybe look into the NIH Model AD program or research into those resilient individuals or the role of the immune system. Dig into the areas that resonated most with you.
There was a wealth of information out there, and the field is moving fast.
Absolutely. Thanks for joining us on the deep dive.